And then I'll do this. Okay, so I want to welcome everybody into this seminar series. And today's talk will be given by Professor Almeida from Sorbonne University. And this will be a joint presentation by both of them. And they'll be giving us a talk on mathematical models and feedback control of population of agricultural pests and disease vectors. So I will just allow us to get their bio sketch, and I'll call on Ms. Fester to read their bio sketch before they start their presentation. Thank you. Ms. Fester, over to you.
Good afternoon, everyone.
Yes, you can go ahead.
Okay, so I'm going to read the bio sketch now. Dr. Almeida received a PhD degree in applied mathematics from École Normale Supérieure de Cachan, now ENS Paris-Saclay, and a habilitation at the regard des researcher from Université Paris et Marie Curie, now Sorbonne Université. He's currently director de recherche, that's a senior researcher at Laboratoire Jacques-Louis Lyons, CNRS, Sorbonne University, and Université Paris, Cité. His research interests include mathematical modeling of disease vector and agricultural pest populations, wound healing and tumor growth in general, mathematical modeling in ecology and life sciences. Dr. Agbo Bidi is currently finishing his PhD thesis at Sorbonne University under the supervision of Louis Almeida and Jean-Michel Coron. Thank you.
Okay, thank you very much, Ms. Fester. So I will call on Professor Almeida and Dr. Agbo to give their presentation.
Okay, thank you very much for the presentation and for this invitation to give a talk at your colloquium. We're very honored by this invitation. I'll start presenting a bit the problems, and then in the second half of the talk, Kala will be taking over and presenting some more specific work with a little bit of numerics, which is maybe more adapted for this seminar. I'll just try to share my screen. Can I? Yeah, I think so. So let's see. Okay, and now if I go on to slideshow or full screen. Yeah, you're seeing my screen in full screen?
Yes.
Okay, fine. So this is some joint work with with Jean-Michel Coron, Nicolas Vauchelet, Alexis Léculier, Amaury Hayat, and Nathan Lichelet, and of course the two of us. There's also some field work done in French Polynesia by Hervé Baussin, Françoise Mathieu Daudet, and in Cuba done by René Gatto and Miss Ladis Rodriguez from Instituto Pedro Curi. So what we will be describing is how to model and use some control tools to manage the populations of vectors of diseases like mosquitoes. That's what we'll be talking most about. But the methods we'll be presenting are also valid for agricultural pests, which have similar behaviors, and we will be giving you some examples of that. So I'm just having a problem with changing my page. Yeah, I'm stuck on the first page for some weird reason. I'll stop sharing and come back just to see if that solves the problem. Okay, share the screen. What is it now? Oh, it's strange that it, oh, here, this one. Okay, let's see if now he lets me change the page. Okay, my sharing is paused. Why is it paused? I'll just try again. Preview, share. Why is my sharing paused? How do I get? How do I unpause my share? There's some weird problem. It says my sharing is paused, but I don't know why. Your screen sharing is paused, and how do I get out of this? Strange, I never had this problem.
Can you try again? Maybe you stop and
Yeah,
share.
stop and start. That's I already did it 3 times, but I'll try still once more. Okay, I choose my preview. I say share, and it puts me some orange message saying my sharing is paused. Really weird. I've never had this problem before, so I don't know exactly what I can do. I'll close my window, reopen it, my PDF window, and see if with that it will. Okay. So this is what I have. I'll just close the preview, reopen it. Opa. Okay, now I go back to the Zoom and I'll ask to share again. So share, preview this, share. Okay.
Excellent.
And now I just try to go in full screen, but it was when I went into full screen that we got into trouble the other time. Let's see. Okay. Annotate current. No, I don't want to annotate. I want to. Okay, good.
Good.
Moving. Sorry for the technical mess. So we'll be talking about vector-borne diseases, which unfortunately is something that Africa suffers a lot from. So what is special about these diseases? It's that you can't give directly the disease from one human being to another one. You need a vector. So in fact, you have three components in this type of diseases. You have the vectors, which are usually things like mosquitoes or ticks or fleas, which bite one human. And it's when they bite the first human that they eventually catch the disease. And once they go and bite another human, they will be transmitting it to a second person. So the humans will usually be the hosts for most of our models. And then the third component of these diseases is the disease itself. So the pathogens, which can be Plasmodium in the case of malaria, and some virus in the case of dengue, chikungunya, Zika, or yellow fever, or even some bacteria like for the Black Death. We will be mostly talking about arboviruses, so things like dengue, chikungunya, and Zika today, and you will see why. So, as you know, they are really the most dangerous animal for mankind, and they're transmitting lots of diseases, mosquitoes. Here we're seeing the mosquito life cycle. We'll be coming back to it later. And essentially, different types of mosquitoes. You see my pointer or not? When I move the arrow on the screen, do you guys see?
Yes, yes.
Okay, good. So different types of mosquitoes will be transmitting different diseases. So I think where you are, it's essentially the Anopheles mosquito and malaria, which is the biggest problem. Here in Europe, it's more the Aedes type mosquitoes, which
Sparkle your
are the
phone.
problem now because they are invading Europe and they transmit a lot of diseases that we were not used to having in Europe. And that's what we're seeing here, is that by now, most of the south of Europe has been invaded. That's the zones in red by this Asian tiger mosquito, which is called Aedes albopictus. And so these diseases, which until 10, 15 years were not a problem in Europe, because even if someone arrived with dengue from elsewhere in the world, especially tropical areas where it was very widespread, the person could not transmit it to other people here in France. So we could never have an epidemics of dengue in France until a few years ago. Nowadays, it's no longer the case, and that's why we have been investing also a lot of effort, and France is starting to react and to try to fight back on this danger and find ways of controlling this risk of having arbovirus epidemics. And in fact, the French authorities even made a report just a few months ago, judging that there's a fairly high. So here you're seeing the presence of the mosquito, the Asian tiger mosquito in France, in the beginning of last year, so in January 2024. And you can see that it's nearly all over the territory by now. And and so the risk of having an arboviral disease epidemic, so which are diseases transmitted by this mosquito in mainland France, is fairly high. So they judge that we have a fairly high risk of having such an epidemic in the next 5 years, which is becoming an urgent problem. And if such an epidemic happens, both the vector control system in France are not prepared for it, and the healthcare system is also not prepared for it. So we will be having big trouble, maybe as the ones we had with COVID in the past. So, so we are trying to use our math to help prevent that from happening and to propose solutions on how you could fight that type of danger. And the advantage of vector-borne diseases is that if you remove the vectors, they can't spread. And that's why it wasn't a problem in mainland France until very recently, is because we didn't have vectors for these diseases. And so that's an advantage compared to diseases like the COVID that you can transmit directly from one human to the other. The disadvantage is that even if you do some lockdowns and you keep people staying in their homes away from each other, the mosquitoes can still come to bite them and transmit the disease. So the way you fight COVID or the way you fight a vector-borne disease are not really the same. And so the methods we will be exploring today are methods for fighting vector-borne diseases, or as we will see, some agricultural pests which work the same way. So normally the ideal way of fighting them would be to do some natural and mechanical control, which essentially goes through trying to prevent them from having good conditions to reproduce, or have natural predators that could fight that population. But the problem is that is very time consuming, and you need your population to adhere very strongly for that to be efficient. But it's also always one of the things that you try to establish. Then what people have been using the most to control this type of problem is insecticides, by trying to kill the mosquitoes or the ticks or whatever animal is the vector that you're wanting to fight. The problem is that has very strong ecological drawbacks. Uh, it's not species specific. So normally the insecticide, it will kill the mosquitoes, but it will also be killing lots of other insects that are useful, like bees or other insects which pollinize your cultures. It will also have an indirect effect on the predators. So you will be killing also the predators, which means that when the mosquitoes come back the next year, you'll have less predators in your environment to control them. So you will even have a bigger mosquito problem. And then there's something which happens a bit like for antibiotics, is that you have resistance to the treatments that develops very fast, because mosquitoes have generations every 2 weeks. So you can have some selective pressure put by your insecticide, it will just select the strains that are resistant to the insecticide, which means that you'll have to put more and more insecticide to fight them. And so you will just be aggravating all the other problems of using the insecticides. And or you will have to change insecticides, but again, you don't have 1,000 different options that you can change, and they've really developed resistance very, very fast. So people have been investing on biological control techniques, and that's what we'll be talking about today. So essentially, we focus in our work on two methods. One is the Wolbachia method, which is the method that is put forward by the World Mosquito Program, where you try to replace the population of your wild mosquitoes that transmit the disease by a modified population of the same mosquitoes, which are infected with a bacteria called Wolbachia, and which has the effect of making them less efficient on transmitting the disease. But what we will be talking about today is another technique, which is called the Sterile Insect Technique, which is a lot less specific of mosquitoes and which is also used in agriculture, as we will see. And there, what you're doing is that you're releasing big quantities of sterile males. By crossing with the wild females, they will make that the wild females that cross with them will have eggs that don't hatch. And so you remove potential individuals from the next generation by doing this release of sterile mosquitoes. So since it's targeting reproduction, it's very specific to the species because by definition, it's the individuals that can reproduce with the mosquitoes that you are releasing that are touched directly. And so even if you're intervening, like here in Europe, against these Asian tiger mosquitoes, you will not be destroying even the population of our classical Culex mosquitoes because they don't reproduce between either themselves. So it's really something which allows you to target very specifically one species and not bother the other ones, which is a very big advantage compared to insecticides. And the bad thing is that you need the recurrent intervention, as we will be talking about. And that's why we're developing all these feedback control techniques to try to be able to do these subsequent interventions at a very low cost, which allows us to keep these interventions going for the long run and keep the environment free from mosquitoes or from the mosquitoes that transmit your disease. So this is just is a diagram of your mosquito life cycle. When they're a problem is the adults. In fact, it's even the adult females because those are the only ones that bite humans, and so the only ones that are really vectors of the diseases. They bite because they need blood in order to be able to lay their eggs. So every time they need to lay their eggs, they will have to have what we call a blood meal. And it's when they have this blood meal that they will be trying to get some blood from the person they're biting. And at the same mute. Okay, I'm unmuted. You listen to me now? Yeah. Okay, I was telling you that it's the female adult females the problem, but finally knowing all about this life cycle gives us other ways of also attacking the mosquitoes eventually through predators that can attack the larvae or through removing the places where they can lay the eggs, as I already said. So it's good to keep it in mind. And so you can start by doing models where you take into account the whole life cycle. And to do that, I think I lost. It's because For something, I lost my possibility to change screens again, but I think I understood it when it's open now. How do I? No. It goes into some annotate mode, and that's, I think, is what blocks my. And how do I get out of the annotate mode? I don't want the annotate mode. I want to.
So
Okay,
maybe you can
good. It's
move.
moving. So I was telling you from what you know about their biology, their life cycle, you try to make a diagram to tell you that females lay eggs, which after some time become larvae, which after some time become pupa, which can either give rise to a female or a male, and then put all the death rates for all of those things. And once you have this diagram, and with the extra information you have about your mosquito life cycle, you can write a system of ODEs to describe the dynamics of this population. And then, as we will see, a system of PDEs when you also want to take into account space in your problem, or if you wanted to take into account some structure in your population, like resistance to the insecticide, you could also write PDEs for that. But that's not what we'll be talking about today. So essentially, from the diagram, you write your system of ODEs, and this is the dynamics of your native population. And then you want to see how you would be modifying your system to take into account the fact that you have a control on that system, which is the possibility of releasing sterile males. So this is the sterile insect technique. And essentially, it consists on mass rearing mosquitoes, which are the same as the ones you have in your field. Then you separate the males from the females, and you just irradiate them at a sufficient dose to make them sterile, but low enough not to affect their fitness so that they're still competitive with the wild mosquitoes when you release them into the wild. And then you just release them either by traditional methods where you just go around with buckets of mosquitoes and release them, or nowadays using drones, which allow you to do a lot more precise way of implementing a release strategy. These mosquitoes come and cross with your wild females, and the eggs which will be born from that type of mating will be sterile and so will not contribute for the next generation of your population. And the only thing you have to do is you just go to the system you had, you add one new compartment, so one new population, which is your sterile males. The only way you have of producing sterile males is by releasing them. So this U here is your control function. And the only thing they do is that they will be dying at a certain rate. And then you have to see what having sterile males changes in the dynamics of your system. And essentially, it changes by the fact that when you have one female that crosses with a wild male, you will have an egg in the next generation that will be hatching. But when she crosses with a sterile male, you will have an egg that won't hatch, and so which will not be counted in this E population here. And so essentially, you need to take into account what is the probability of your female to to mate with a wild male. And essentially, that will be something like the number of wild males divided by the number of wild males plus sterile males. So the more sterile males you put, the less is the probability that your female will cross with a wild male. And so the probability that it will have an egg which will hatch is going to zero when the number of sterile males is going to infinity. That's essentially how this technique is working. And then you can put some coefficients to take into account the fact that maybe there is a preference between wild males and sterile males. Since they were irradiated, they could eventually be a little bit less fit. And so this gamma here would be something smaller than 1. And also you need, especially when you have a very small population, since you have sexual reproduction, you need a female to find a male. And so that will depend on the female searching efficiency. And that's what this parameter alpha is here. But in big population, it's not even playing a big role. Okay, so now you have a problem which is really a mathematical control problem on a system of ODEs, of population dynamics. And that's what you really will be having to do and developing some math to see how to control this in a smart way. So just to mention that this technique, in fact, it's being applied to mosquitoes recently, but it's an old technique that from the biological and field work, not for the math, but it was invented for the screwworm fly in North America, and we'll be talking a little bit about that. But it's also used against fruit flies, which are attacking lots of different orchards in Europe and elsewhere in South America. It was used in Africa, in the Gulf of Guinea, against the tsetse fly. It's being used against moths, and recently it started to be used against mosquitoes. And generally, what the FAO and the IAEA is involved because this is the International Agency for Atomic Energy, and they are involved because they are the ones who are providing the possibility and the tools for sterilizing the mosquitoes, especially in low-income countries which are not able to buy those tools themselves. The IEA is helping a lot. It's the case of Cuba in particular, where it's them who are really financing all the irradiators and all the deployment of this technique. So, and they say it's a very good return on investment. So it's a technique that has been used since the late 50s. So by now we already have 60 years of experience with this technique. And as you will see, it has had some very striking successes. So this leads us to think about the problem of the spatial invasion, of how these mosquitoes are going to move in space. And here you have a diagram where you're going from left to right and top to bottom. You're moving in time between 2004 and 2022. And in red, we're marking the zones where this Aedes albopictus, so the tiger mosquito, was detected in France. And you can see that it came through the southeastern border because it was already present in Italy. And progressively, as the years go by, it has been invading France from the southeast to the northwest. So you're led mathematically to think about propagation of invasive fronts, which is something that has been studied very in big detail in ecology for many other species, and which even mathematically is a very well developed theory as an abstract theory in PDE. So we will try to do models to describe this invasion and to describe how control theory can help us push it back so that we can protect populations from these disease vectors. And this is the original problem where the sterile insect technique was developed. And it's also a way for me to illustrate what is a rolling carpet strategy. So these were flies that were attacking the cattle in the south of the United States in the 50s. And they had the idea, since they realized that when they irradiated these flies, they became sterile, they had the idea of releasing mass quantities of sterile males. And they started doing that in the late 50s in the south of the US. And by doing that in quite fast, so here you're seeing it's a 2-year period, they got rid of those flies in all the south of the US. These flies, the problem is that the larvae, they put their eggs next to wounds or to warm iron markings of the cattle, of the cows, or of the sheep. And then the larvae of these cows, they just dig into your flesh, and by doing so, they can give rise to some inflammation. And the cattle, they were having very heavy cattle loss at the time. That's why they developed this technique, and they did a very strong investment. But as you can see, they just deployed it all the way from the Atlantic to the Pacific. So if you have the money and the means to do it, you can deploy it in very, very wide areas, so at the scale of a continent. And then what they did is that the US got the idea, if we can push them a little bit further down, that will give us some barrier area to avoid having some reinvasion by these flies. And the Mexicans also hopped into the program and started releasing these flies in Mexico progressively. And so as the years went by, they started releasing them progressively further and further south until by the 2000s they had reached Panama. And that's a very good place to put a barrier because since it's very narrow, you need a lot less flies to put a barrier at the level of Panama than if you try to do it at the level of the south of the US. And so since the 2000s, they have been doing what they call a permanent barrier of releasing sterile males in this area so that flies from the South America are not able to reinvade Central and North America. And at some point, it would be good to attack the flies in South America. The problem is that you need the money, and you have Amazonia and the Andes here, which are natural problems, natural barriers for deploying the technique. So at some point it should be done, but I don't think it will be right now, especially because right now the only ones who have the money is the US, and they're not really very nice at aiding other countries to solve their problems. So let's hope someday that will be done. But from the mathematical point of view, how do you write that? Essentially, you describe the propagation of your mosquitoes in space as a first approximation by a reaction diffusion model, where you have some term which is linked to the fitness and the reproduction of your mosquitoes. And then you say, okay, as they were doing for the screwworm, I will be acting with my sterile insect technique in a band between 0 and L. And in this band is where I release my sterile males. So it's where I'm acting on the system. I can also do the same model for using some killing action on your mosquitoes, which can be just capturing them with some traps, or which can be using an insecticide. So the same type of math can also be applied for that case. But here we will be more talking about the sterile insect technique. And what happens is that you have your population that when you do nothing is propagating in space. That's what we're representing in this line at some speed, C0. So it's a traveling wave solution of this system of reaction diffusion equations. And when you introduce this action, if it's strong enough, we can prove mathematically that it will stop the propagation of your wave. But the rolling carpet strategy is even better than that, is that instead of just stopping it, you start moving the area where you act towards the place where the mosquitoes or your pest is arriving in general. And that way, by doing so, if you have a sufficiently strong action, you can actually push back the invasive wave. And that's what we're representing graphically here. And so then it becomes a mathematical problem, and it becomes a control problem because your control is exactly the action that you're implementing in this area. And then you can even make it into an optimal control problem to see what would be the most efficient way of acting. But even if you're just worried about finding one way of acting that works, is already a very interesting mathematical problem that we studied. And we were able to prove actual mathematical theorems on that system that we published 2 years ago with Alexis Léculier and Nicolas Vauchelet. And essentially, it's doing what you expect it to do. Is if you don't have enough mosquitoes, then you can't push the invasive front back. That's what one is saying. Two is saying if you have a number of mosquitoes which is above a certain threshold that you can compute, then you can push your invasive wave back. And that if you want to act in a very small band, then you need to have very high density of mosquitoes in that band. If you want if you allow yourself to act in an infinite size band, you will still need more than zero density of mosquitoes to do it. And if you want to move very fast to push back your front, then you need many mosquitoes. So the more mosquitoes you have, the faster you can push your front back. And you can also do the numeric simulations of that. So, and that's what we're seeing here. What you want, so in the vertical axis, you have time. In the horizontal axis, you have space. You're intervening between the two red lines, and you're trying to push your intervention zone to the left, so against where the mosquitoes are coming. So high density of mosquitoes is yellow, low density is violet. And what we are seeing here is that if you have a certain quantity, which here is normalized to be 3.3 of mosquitoes, and you try to move at the speed, so the speed you're moving is essentially the slope of your red lines, since this is time and this is space, then you can see that it seems to work, but after a while you start having mosquitoes at the back of your intervention area, so your intervention failed. But if you increase this quantity of mosquitoes just by 10%, you can guarantee through the type of results as the ones we proved in the previous theorem that you will never have a reinvasion on the right of your intervention area, so that in fact you can push your invasion front back. And that's something that they knew from the field, since they had succeeded in pushing the screw worm all the way down to Panama, but they didn't know exactly how to optimize that. They hadn't done the math for it. But in fact, the math is perfectly doable, and it's even quite interesting. And so now that we did the math, we have been working on how can we really go from the math to the field applications. And so there had been our collaborators from French Polynesia have been intervening in a small island in the atoll of Tetiaroa with the incompatible insect technique, which for math, it's the same thing as the sterile insect technique. And they, as you can see here, these populations fluctuate along the year. And in blue is how many sterile incompatible insects they were releasing at some point from 2015. And you can see that by releasing these sterile mosquitoes, they were able to push the population all the way down to very, very low level, a level where they were not capturing mosquitoes at all in their traps. And that lasted for nearly 2 years. But since in the meantime they stopped intervening, then the population just comes back. That's, as I told you, the problem of this technique is that if you stop intervening, then at some point your population goes beyond its, even because people by traveling can bring mosquitoes in. You can have some eggs that were left and that can eclode a long time after. And if you go beyond the threshold for the viability of your population, then your population just has a natural tendency to grow exponentially towards its carrying capacity. And then the fact that it fluctuates, it's because with the weather and with the seasons, you will have that some seasons are more favorable or less favorable for the mosquitoes, but you will be oscillating at a high level of mosquito population. And so you will have a risk of having an epidemics of the diseases they transmit. And this backstepping control technique is essentially to try to find good ways of intervening so that you start by intervening with a lot of mosquitoes, but with time, you can keep your mosquito population down while using very few mosquitoes. And that's what I'll leave that for Carla to explain you more in detail, the results we have for that. But just the projects we have been doing. So we have some agroecology products. So how to use um, intervention in field cultures to avoid using pesticides and control pests. We have one big project that was approved just now on how to prevent the dengue outbreaks in mainland France, which will be running for the next 5 years. And then we have these projects with the people in Polynesia and a similar one with the people in Cuba, and now also with people in south of Portugal, where these are projects that are financed by the World Health Organization and by the International Atomic Energy Agency, and which are for getting proof of concept that this technique works well against the mosquitoes in the field. So in this case, in this project concerns Tahiti, the Cook Islands, and the Easter Island, and that's something which is ongoing. And what do you do in this project? So you're trying to prove that really you have one area where you just do standard interventions. That means just removing the egg laying sites to try to avoid the population of mosquitoes to grow easily. And another area where you intervene with your sterile mosquitoes, and you try to show that in the area where you intervene, you can get a lot better results than in the area where you don't intervene with the sterile insect technique, as what they had been able to do in that small island in Tetiaroa. And once you have that, so this is what is being implemented now until 2026. Once you have that, the idea is to move on from that. So this is the island of Tahiti. Papeete, which is the capital city, is here. And we are starting to do it here in this area that we saw in the previous diagram. And once we succeed in doing in this area, what we will do is that we will be intervening with sterile insects in the yellow area, so further and further towards Papeete. And at the back, we can't stop intervening because we know that if we stop intervening, there's a risk that the mosquitoes can come back and the population can grow. But we will be establishing this feedback control type of technique where we monitor the population of mosquitoes and we intervene in a way which is adapted to the number of mosquitoes we observe. But since we have pushed the population close to zero, that means that we will be observing a small amount of mosquitoes. And so if we intervene in a dynamic way before they can grow, we can keep them at a very low level with very few mosquitoes, as Carla will be showing you. And then you just go on doing that. You go on moving. So it's the rolling carpet strategy. You're trying to push, to construct a wave that pushes back the mosquitoes. So you intervene further and further along in the yellow areas, and you do feedback control in the green areas until you go all the way through the Papeete urban region, so that you will be protecting all this population from dengue or other diseases that are transmitted by the mosquitoes, which in that case is Aedes aegypti mosquitoes. And that way you can protect the half of the Tahiti population in a few years just by deploying this type of strategy. So now I'll give the hand to Kala, who will be showing you what we have been doing for this feedback control. But the important thing is you need to be able to control the green areas with a few mosquitoes so that you can keep many mosquitoes to attack the area where the mosquitoes are still very numerous. And for a small island like the Motu we saw in Tetiaroa, you can attack the whole island at the same time. But even for an island like Tahiti, and even more so for a country like France or for a continent, you'd never have enough mosquitoes to attack everywhere. So that's why it's important to do rolling carpets, is that you start at some point where you have enough mosquitoes to treat that area. And once you treated that area, you go and treat a little bit further away just by keeping your feedback control in the area that you have already freed from the mosquitoes. And by doing so, progressively, you can free the entire zone and do an area-wide intervention. Okay, so I pass the talk to Kala, who will be, so I stop sharing, and Kala should be able to share at my place.
Yes, thank you very much. While Kala shares his screen, I want to thank you very much for the very enlightening talk. And yes, so I'll just wait for Kala to share his screen.
Okay.
So that it can continue. Yes, Kala, over to you. Thank you.
Yeah.
So do you see my slides?
Yeah, we see them.
Yeah.
Yes, you can see
Okay.
your screen.
Thank you. Thank you for the invitation. I present here the feedback control results for the SIT control system. So more quickly, the SIT ODU control system that Luis presents in his presentation is the following one. And the problem here is to design a control such that the population, goes to 0. So I will recall the states of these dynamics. So here E is the density of aquatic phase, and F and RM is the adult population, and MS is the sterile male population, and U is the density of release. Okay, so beta E and delta are the life parameter of the systems. So here beta is the oviposition rates, and delta are the death rates of the population, and nu E is the hatching rates. So the population viability in the absence of controls is determined by these constants denoted here by R, which when this constant is less than 1, is smaller than 1, the population goes to 0. Okay, so we suppose in this study that R is greater than 1. So we have the population at some positive equilibrium. So one of the problems that Luis presents is the resolutions of the population when we stop the release. Okay, for example, we consider the control given by relation 2, which consists of releasing some quantity of sterile in the population, as long as the density of the X is greater than 0.1. And from there on, we stop the release. So here is the numerical evolution of the population. So I will present it more later in the presentation. So when we stop the release after some times, okay, in the reds, we have the evolution of the control. So after, when we stop the release, the population come back after some times. So the global, the feedback control and the stabilization is to construct a control that maintains the population to 0. So in control theory, this notion is the global asymptotic stability. And here, our equilibrium point is 0. So mathematically, we must prove this property. So the first property is the local stability, which is when we start from an initial point close to 0, the the trajectory of the population also is close to 0. And the second point is the attraction of the global attraction of 0. Okay, so our goal is to construct a control law which depends on the state of the system and that ensure the stability of 0 asymptotically. So we reach this objective by these two controls, okay, given by relation 5 and relation 6. The first one depends on X and the total number of the mosquito, the male mosquito. And the second one depends on the white male density and the X density. We have also some feedback law, some linear feedback law, depend only on the white male or total male. So the stability results in present in this paper. So numerically, we use the explicit Euler method to approximate the numerical solution. So the equation, the equation that gives the numerical solution is given by relation 9. And when we plot our system with this feedback law, we have this evolution. So in the red line, we have the control, okay? And one can see that when the population also is small, the control is small. So this control, when we apply this control, when the population is small, that avoids the resolution of the population and also reduce the cost of the control. Okay, we have also some nonlinear control that we construct. One can find it in this paper using a backstepping method. So these control theory tools. Okay, so in this paper we discuss about this nonlinear control law. So one of the challenges for applying these feedback laws is to measure the states we need to compute the control law. So here, when we release in the population a sterile, we have different kind of mosquito in the population. We have a female that mate with a sterile male. We have those female that mate with a white male. We have a white male. We have sterile male also. So to get the measurement, for example, of a female that mate with sterile male is very costly or very difficult. So to solve this problem, we construct another auxiliary states, another auxiliary dynamic called in theory, in control theory, an observer system that use only the measurement of white male and sterile male to estimate the total state of the system. And another way is to design a control using a reinforcement learning based on the observation of a total adult mosquito. Here is a total male and total female. Okay. So, for example, I'll present here the process for a reinforcement learning control design using an observation of total male. So, we discretize the equation to create a simulation environment, and the control agent acts in the system based on the state, based on the observation and the reward in minimizing an objective function. So here, every 7 days, the control agent gets an observation which here is total male, and also an reward in minimizing an objective function. And he designed an action every 7 days, and this action is used to train the system, and he repeats the action, okay? And the control agent find himself the action policy that is the optimal to minimize the dynamics. So this gives this figure, and here U is the action, okay, the action of our control agent, and that asymptotically stabilize our system in 6 months of observation. So we also consider the spatial movement of our population by a reaction diffusion. So here we add Laplacian operator which model the spatial movement of a population, and U can be a vector, okay, and F is the interaction of a population like reproduction or growth or death in the population. So, for example, that is the movement of an insect in an area, okay. So the Laplacian operator model the spatial dispersion of an insect population. So for numerical simulation, we use a central differential chain to approximate the second derivative. Here is one-dimensional. Okay, you can also do it in two-dimensional, and the time stepping of the solution is given by relation 14. So to implement this scheme, we need some stability condition, okay, of the time step and space step given by relation 15, and also the reaction function must be Lipschitz. Okay, you can also add some boundary condition, okay, such as Dirichlet or Neumann, okay, based on the problem context. So the SIT PDEs that we can consider, so to model the dispersion of mosquito population, is the following one. Here the X dynamic as X not move in space, so we don't add the Laplacian operator for the X dynamics, but we add this to female, as female adult female move in the space with a diffusion coefficient noted here by T1. The same for male density, sterile male density, in which there is a control. And for boundary condition, we consider Neumann boundary condition. That means the adult population don't leave our areas, okay? So this boundary condition biologically is that our adult population don't leave the domain we consider. So for feedback stabilization, this law, okay, we prove in this paper that this law ensures the global asymptotic stability of the system in the whole domain, okay. So for the problems such as well-posedness, also we present this result in this paper, okay, as we don't have more time, so I not present them in this presentation. So numerically, we choose as carrying capacity the following function, okay, with the domain given by relation 22, and this give this figure that we plot in 2 dimensions, okay. So we have the X density for T equal to 10 days, and when we apply the feedback law, we have the repartition of X for T equal to 200 days, okay, in the whole domain. So this is the end of my presentation. For those who are interested to this work, we have some references here. I forget to, for reinforcement learning, the code is available in GitHub. So for those who are interested, they can also find this code and plot it in their computer. Okay, so thank you for
Okay.
Yeah.
Thank you very much, Dr. Luisa Melda and Dr. Agbo Bidi for this wonderful presentation. In fact, I know that there are a lot of people that must have taken one or two things, from this lecture, and I know that many people have learned, but also I know that people have, some questions to also ask, so I will take reactions now. If you have some questions, maybe you just have some questions.
I have a question.
Oh, okay. Okay,
Can I go ahead? Hello?
yes, please, you can go ahead. You can go ahead, ma'am.
Thank you
Hello,
very
yeah,
much.
you can go ahead, please.
Okay, thank you very much for this talk. It's very enlightening. I just have a question about the carrying capacity, because you have a function that defined the carrying capacity. I'm just curious as to what was the motivation and for defining such a carrying capacity.
Is to approximate the heterogeneous of the space, okay? So, for example, if you have some water in some part of your area, water, mosquito population usually use water for their reproduction, is normally to consider that in this part of the domain, the density will be very high there. So that is the heterogeneity of the domain. Okay, so there's no motivation.
Okay.
The function we used is not coming from any data. That was just getting free Gaussians of different heights just to show that the method works. But
Okay.
you can then also use it with real data. But because normally these mosquitoes, on the one hand, as Kala was saying, they need water to reproduce. That's one of the ways of attacking them, is to try to remove the water from the flower pots and all of that. But they also need, especially Aedes aegypti, they only live very close to humans because they're really very specialized on attacking humans. And so you will also see that their presence will be correlating on the one hand with places where they can reproduce, and on the other hand with places where there are humans available. And so it's natural, if you really want to do realistic stuff, to move from the ODE setting to the PDE setting if you want to take that into account.
Okay, so in defining the carrying capacity, you took into account the dimension of the domain where you would find the mosquitoes. Is that how you went about defining the carrying capacity?
Yeah, essentially, as usual in a population model, it's measuring the maximum population that your environment can sustain at that point in space. Of course, you don't get that precise information, but you can get from the field this type of information that in a certain area they can reproduce more easily than in another, and then take that into account when you try to develop your strategy to fight them, because then you would also be normally having to release a lot more sterile males close to the places where they will be reproducing a lot than close to places where they're not that present and where your sterile males would be less useful to be.
Okay, thank you. I have another question, but I'm going to let others ask, and if not, I'll come back to my question.
Excellent, Pa. Thank you. So do you have any other question? Anybody wants to ask any other question?
Yes, thank you, sir.
Yeah.
Good afternoon,
yes,
sir.
please, if you want to ask question, just use the reaction button, raise up your hand so that we can call you. Okay, so go ahead with your question, please, Dr. Diko.
Yes, thank you. It's been quite long.
Thank you.
I quite appreciate the speaker. I didn't join too early. So, but a lot of times when we have some numerical simulations, interpretations, and we have the graphs, people just show us the graph, and we most times don't really bother about the interpretation. Yeah, although I see it's a two-dimensional graphical illustration, but I love if he can just explain a bit about the interpretation based on the problem that was solved. Yes, and I don't know why it must be two-dimensional. Is it because you have just a PDE? Why is it not showing a three-dimensional kind of graphical illustration? I just want an interpretation to the graph anyway. Thank you very much. Then I don't know if you have access to the presentation slide and the reference you pointed out. Yeah, we would love to have it. Maybe we should send an email. Okay.
They are promised to give them to me. I'll send to everybody after the. Please, if there's a link in the chat box. I want you to. It's a survey. Maybe you just fill that survey so that we can have your email, so that you can send that the link to this seminar series and the what do you call it, the slides to your email directly.
All right, thank you.
Yeah, we
yes,
can
Dr. Kala, want to react to this question?
I see my presentation is something like 6 mega. I don't know, Carla's one should be maybe more or less the same, maybe a little bit less. So I think it will be a total 10 mega. We can put it on a server, or we can send it to the organizers, and the organizers put it on a server in Nigeria, and then you just come and download it. That way we don't have to send very heavy mails around. It would be more efficient, maybe.
Yeah, I think that would be best.
I'll send the link right away to you, and then you can recover it, and either you use the same link, and I keep the thing in my CNRS server, or if you want, you put it in a server in your department, and then people just access it from Nigeria. I don't know, as you want.
So maybe I'll send you an email, then you send me the link.
And I send you the link, and then you take care of that.
Exactly,
okay.
exactly.
That's fine for me. Regarding the question, maybe Carla wants to answer. The 2D is because normally if you have information about where is water, where's a house, you have it essentially a 2D information. That's what you get from satellites. But nowadays it's starting to be possible to get this LIDAR information, where you can even get information on the vegetation all the way from the ground level to the canopy. And if you have that type of information, you can develop a 3D model also and study the 3D model. The delicate thing about the 3D model is that somehow you can suppose that your mosquitoes don't have a preference between moving in the X of the Y direction. But if you add the Z direction, Z will have gravity working against them. So your diffusion operator will surely have to take that into account, and maybe other things. So then you need to get more information about how mosquitoes fly. You can't just put a three-dimensional Laplacian with the same diffusion coefficients along all directions, because I don't think it would be realistic. But if you have the good information, you can work in 3D, and it would make sense. I don't know if Carla wants to add something.
So I think is enough for the.
Okay.
Yes.
Any other question? Okay, Professor Augusto.
Yes. So my question regards the coefficients for the female preference that Louise mentioned earlier. You said something about if that is small, if that is small, doesn't that imply that the total number of eggs being laid will be small overall if the female have preference?
So if the females have a preference for wild males, you are a bit in trouble because then, for instance, if a sterilized male is having a chance 100 times less of mating with a female than a wild one, then just to make up for a wild male, you need to have 100 female sterile ones, and still half of the time your female will still mate with the wild male. So that gamma, you have interest in having it as big as possible because it's measuring how competitive your sterile males are relative to the wild ones. The wild ones are the ones you don't want them to mate with the females because when they mate with the females, you get mosquitoes in the next generation. And you expect that when you irradiate mosquitoes, you will be reducing a little bit their fitness. So if you do nothing else, you expect that that gamma will be smaller than 1. But in practice, people have optimized their radiation doses so that it is as close to 1 as possible. And they can get things like over 0.9, 0.95 in field experiments or in lab experiments. And the IAEA has been optimizing exactly the level of radiation because you know that the more you irradiate, the more sure you are that your mosquito is sterile. But if you make mosquitoes that are completely sterile but can't mate with any female, they're useless. So you need to find a good compromise between the two. And for things like the tsetse fly in Africa, they could even make gamma bigger than 1. And how do you do that? You have to somehow cheat. They were either putting their wild males that they were releasing, they were putting pheromones on them so that they would be attracting more females, or they were feeding them with very high protein diets so that they would be particularly strong compared to the wild tsetse flies from the field that didn't have those advantages. And in that case, you can even make the gamma bigger than 1. So you really want gamma to be as high as possible because that will increase the probability that your female will mate with one of these sterile males, and so that you won't have a progenitor.
Okay, well, thank you. Yeah.
Okay, so I have a question. So I think this question may be to Kala.
Okay.
So I realized when we were trying to discretize your partial differential equation, you developed even in the ordinary differential equation, you used the forward Euler, and when you also discretized the partial differential equation, using the the central difference formula for the Laplacian, you also use the forward Euler, which is the forward time center space. I'm thinking in discretizing the PDE, in solving the PDE for the time stepping, you actually gave the stability condition, which I feel that is quite correct. But don't you think that there are some higher order numerical method you could use instead of just using the first order time stepping scheme?
I think it has to do with the convergence with the optimal control.
So numerically, as our objective is control, feedback control. So numerically, we just compute some chairmen and some codes, so to have a numerical evolution of our system. So we don't perform the numerical simulation to our system. So
okay, so you see, my question is, how are you sure that the first order scheme you are giving is actually giving the actual dynamics or the evolution of the system you're actually looking at? If and also using the first order scheme, don't you think it will take some time due to the time restriction that you're giving in satisfying the stability condition you just gave?
So when we consider your domains and some time step, you have to choose the time step such that this condition, okay, holds, and then you have your evolution of the equation. So I don't know if I answered.
Yeah, you answered rightly, but I'm just saying that suppose the time step, if the system were to be very stiff and the time step you choose get to be, you have it will restrict the time step to be very, very small.
Yes.
Yeah, so I'm saying if you try to run the simulation, it will increase the time, it will take for you to actually complete the simulation. I'm saying instead of taking much time, why don't you use some order schemes that are stable, that will also generate those schemes, but you can take a larger time step.
Okay. Well, I'm not a numerical simulation.
Okay. All right. Any other question? Anybody? Any other question?
How does it sound?
Please, do we have anybody that has any other question to ask? Okay, if there's no question, let me take this opportunity to thank SIMPA through Professor Sidi Kaba for helping us source for the speakers of today's seminar series. And I want to thank the director of NMS, National Mathematical Center, for also supporting us in this seminar series. I would also like to thank Professor Odekunle, who has been my support also, and Professor S.A. Okunuga, my supervisor, who has mentored me and taught me how to do research and how to do this thing, coordinate and do everything mathematics that I know. And I want to also thank Professor Augusto for her support for me. In fact, he has always helped me and also always told me and told me things and giving me ideas of how to advance my research and everything that I do. And I want to thank everybody for coming for today's seminar series. Next month, we are going to be having a talk from Dr. Mufta Rufai from the Polytechnic Bolzano something from Italy. Will be taking us through some talk on numerical analysis. And I want to thank everybody for coming. And I don't know, right? In the last seminar series, I talked about a new section that we'll be having, a new segment we'll be having in our seminar series, which I called problem mentoring and collaboration. And I said that I was going to introduce maybe like a 1 minute or 2 minutes, a 5 minutes opportunity for anybody that has any challenge in his research and want to share with us if he can get assistance from any of us in any of the participants in this seminar series. So I don't know if really you have a problem or a challenge that you have prepared in a slide, a three slide, and you want to share today to us, if you can get help. Is there anybody in this category? Otherwise, we just call it a day. We have no light in Unilag, so I'm praying that my laptop does not die. It's almost getting off. So is there anybody here that has any challenge in his research, maybe your PhD research or your master's research, you have a problem and you have prepared a three slides as I instructed and you want to share with us, maybe you are looking for somebody to help you, give you direction or help you in your research. Anybody? Okay, and if there's nobody, maybe I just call on Professor Odekunle to give us a word of support if he's around, if he's still here. Is Professor Odekunle still here? He's not here. Okay. Is Professor Augusto still here?
Yeah, I'm still here.
Okay, just give us a word before we go. Thank you.
Okay, a word, A. A word clown. Well, thank you for putting this together. And I like the idea of asking the students in case they need help to share whatever issues that they're having, because you never know who will be in the audience that will be able to help guide and give direction. And thank you for doing this. This is really good.
Thank you very much, Ma.
Mhm.
Professor Almeida, would you like to say anything before you leave?
Well, I'd just like to thank you for the invitation. It's really nice to see that people are moving in Africa and really taking nice initiatives to work together. We will always be happy to help in whatever way we can. So if I will be more useful for the modeling issues than for the numerics, but people like Professor Kaber are very good in the numerics, and I'm sure he will also be happy to help. And Kala, I'm sure at some point in his life he will like to go back. So it would be nice to keep in contact. And from our side, we will always be happy to have contact with you and get news from how this seminar is evolving. Eventually, helping you find some other people in France or elsewhere to give talks. Don't hesitate to contact us.
Okay, thank you so much.
Welcome.
Professor Kabas, Professor CD started with us, but I think he's out. So please help me send my greetings to him and thank him very well for putting you through this today. I want to appreciate everybody that came today. I want to appreciate all the professors and all the students and all the researchers that made today's talk. I'm going to send the link to the video of these lectures, and also I'm going to send the slides when I communicate with Professor Almeida after this talk. So I'll send the slides to our private emails. Like I said, this is not an association. I'm not trying to create any new association. I'm going to send mails directly to emails, and I'll just use the existing platforms to send flyers and remind people of our meetings. And I want to thank you all for coming, and I wish you a very happy or enjoy the rest of the day. Thank you.
Okay, thank you. Bye.
Thank you,
Thank
Doctor.
you.
Bye-bye.
Bye.
Bye.